Many years ago, when scientists first began studying DNA, its function was unknown. However, scientists did know that there was a transformative molecule responsible for carrying genetic information. Alfred Hershey and Martha Chase were both American geneticists, and in 1951 and 52 they performed a series of experiments which proved that DNA was the transformative molecule that carried genetic information. This new discovery was in contrast to the prevailing theory that genes were made of proteins, as there were more amino acids than bases in DNA, so more room for variation. Though nearly a decade before Hershey and Chase, Avery, MacLeod, and McCarty published evidence supporting the theory that DNA was the molecule responsible for transformation. They also built upon the experiments of Frederick Griffith, who experimented with transformation in virulent and non-virulent strains of pneumococcus bacteria. Their experiments used bacteriophages, which are viruses that infect bacteria. Bacteriophages are made of only two components, a protein coat and DNA, both of which can be radiolabeled. Hershey and Chase first radiolabeled the protein coat. The viruses were then allowed to infect host cells. And the samples were centrifuged. The results showed that the radiolabeled protein was still in the supernatant, meaning that it had not entered the cells, which fall to the bottom of the test tube when centrifuged. This meant that protein could not be the transformative molecule as it had not entered the cells. However, when the DNA was radiolabeled, and the virus was allowed to infect the host cells, and the samples were then centrifuged, the results were much different. The results showed that the radiolabeled DNA was in the pellet at the bottom of the test tube with the cells. This means that the DNA had entered the cells and was the molecule responsible for carrying genetic information.